Okay, for our next talk, we have Dr. TODD Whitehead. Dr. Whitehead graduated from the University of Virginia with a degree in civil engineering. Interesting. And he received his master's in environmental engineering from UNC Chapel Hill. He received his PhD from UCB this past spring, in environmental health sciences with Steve Rappaport as his advisor. So Todd is going to talk to us today about polycyclic aromatic hydrocarbons, one of my favorite class of chemicals, in residential dust as sources of variability.
My name is Todd Whitehead, and I'm here from UC Berkeley, the California Childhood Leukemia study, or the other manifestation of that study is the CIRCLE group. So let's see if I can get this right, the Center for Integrative Research on Childhood Leukemia and the Environment. So first of all, I just want to give a brief outline of the California Childhood Leukemia study background as sort of a context for the work that I'm doing. And then I want to primarily discuss this question of what can residential dust tell us about estimating exposures to indoor contaminants. So the background. The California Childhood Leukemia study is a retrospective case control study that was initiated in 1995. And some of the primary aims of the study are to identify genetic and environmental risk factors for childhood leukemia. And there's this sort of understanding that prenatal and early childhood exposures to chemicals could contribute to childhood leukemia. So with the residential dust samples that we're collecting, our interest is in assessing the prenatal and early childhood exposures that the children have received in our study population, and then to try to see if those exposures are related to childhood leukemia. So just a schematic here about the CCLS study design. So we have cases, children diagnosed with leukemia who are enrolled in the study at the time of diagnosis when they go to pediatric oncology centers. And then controls are matched to the cases on date of birth, sex, and Hispanic ethnicity. And all of the case and the control households fill out extensive questionnaires that are designed to ascertain information that might be relevant to childhood leukemia. So there's questions about parental smoking habits and their pesticide use inside the home, parental occupations, characteristics of the residences, and also just general demographic information. In tandem with all this questionnaire information that we're gathering, we're doing some environmental sampling. So we're collecting dust as part of that environmental sampling. And we're analyzing a whole suite of chemicals in the dust, but the things that I'm going to talk about today are listed on the vacuum there. So PAHs, polycyclic aromatic hydrocarbons, PCBs, polychlorinated biphenyls, PBDEs, which Asa spoke about earlier, polybrominated diphenyl ethers, and also nicotine as a marker for cigarette smoke exposure. So the sort of basic concept is that we want to look at the levels of these chemicals levels of a chemical in houses and compare the distribution of that chemical in the case households to the distribution of that chemical in the control households. And if there's a difference between the two distributions, then we'll infer that that chemical might actually be contributing to the leukemia risk. And we've had some success doing that already. This is an article published by Mary Ward and some of our colleagues at the CCLS in Environmental Health Perspectives. And they observed that there was an association between PCB levels measured in residential dust and the risk for acute lymphocytic leukemia. So this is sort of the end goal for the epidemiologists in our study. We want to be able to find observations like these. But for me, as an exposure scientist, the questions that I really want to examine and talk about today are these four here. And this goes back to sort of what can dust really tell us about exposures. So the first question is, are concentrations of chemicals that are measured in residential dust representative of chemical exposures, especially for children? Another question that's of interest for us is a single dust sample sufficient to characterize the contamination that's in a household? So this has to do with the short-term temporal variability of residential dust measurements. And the third question I want to address is, can dust samples collected after diagnosis be used to estimate past chemical contamination or past chemical exposures And this is an important issue for us as a retrospective case control study. And I'll talk more about that in detail. And then finally, if we observe that there is short-term and long-term temporal variability, what are the sources of this variability in residential dust measurements? So in order to answer these questions, we have to do some repeat sampling. So we go back to a subset of the homes that we've done environmental sampling, and if they're still living in the same residence, we're asking to collect a second dust sample from those homes. Originally, we were doing dust sampling using the high-volume surface sampler, the HVS3, with an interviewer going into the homes and actually collecting the dust. But now we've streamlined that process quite a bit, and we actually just have the subjects package and send us through the mail their own household vacuum cleaner dust. So it makes it quite a bit easier to collect the samples, and it's really a nice advantage of using dust as a media for measuring exposures. So let's start answering these questions. First question. I want to answer the first question on sort of a chemical by chemical basis. So first of all, are concentrations of PBDs measured in residential dust representative of exposures to PBDEs? I would say yes, that they are. So people have shown that the levels of PBDs in residential dust are correlated with matching levels of PBDs in blood, both in serum and plasma, and also PBDs in breast milk. And there's a figure there on the right that essentially shows one of these relationships between BDE 4 to 7 measured in residential dust and BDE 47 in maternal plasma. So as you've already seen from ACE's presentation, BDE 47 is one of the primary congeners in residential dust in California. It's one of the congeners in the penta-BDE mixture. And you can see in this case that the levels in the dust are nicely correlated with the levels in the plasma. So there's some evidence that the dust levels are relevant to biological exposures. Another way of answering this question is by looking at the relative contribution of dust as an exposure route. So this is going to be especially important for young children who spend more time crawling around on the floor and also have hand-to-mouth activity. So in young children and in areas of the world where PBD levels are particularly high in residential dust, such as in North America and also in Asia, the contribution of dust as an exposure route to total intake of PBDs is pretty high as 90% in some estimates. So essentially, you can say that for young children, dust is the primary route of exposure to PBDs. So it's definitely biologically relevant. And then finally, people have also seen that biological levels of PBDs around the world in different populations sort of mirror the global levels of PBDs that were measured in residential dust. So people in North America have higher levels of PBDs in their dust, and they also have higher levels of PBDs in their bodies. And people in California have higher levels of PBDs in their dust than do other places in the US, and they have higher levels, according to N. Haynes, of PBDs in their body as well. Okay, so I'd say that yes was the answer for PBDs. How about for nicotine? Are concentrations of nicotine measured in residential dust representative of exposures to cigarette smoke? Well, again, there have been these correlations that have been observed between residential levels of dust nicotine and matching levels of urinary cotinine, a metabolite of nicotine, in children who have been exposed to cigarette smoke. So again, this is a plot that's showing nicotine measured in house dust and then the urinary cotinine measured in children. And you can see that there's some sort of positive correlation between the two measures. And again, this is in children, not in active smokers. So these are kids that are just exposed to passive cigarette smoke, and still the levels in their dust are related to the levels that are in their bodies. Even more surprising, children who are living in non-smoking apartments actually have elevated urinary cotinine levels when their dust nicotine levels are higher due to past smoking. So children that are living in apartments that were formerly occupied by smokers, but now their families and themselves, they're not smoking, they still have elevated levels of urinary cotinine because of this residual contamination from cigarette smoke. So this sort of suggests that dust is certainly a biologically relevant pathway for exposure to cigarette smoke constituents. Of course, it's a minor contributor to overall intake of cigarette smoke constituents compared to active smoking. But but it could still be an important route for exposure in young children. To move on to PCBs, there's less evidence that PCBs measured in residential deaths are representative of biological exposures. So there has been one study that's shown levels of PCBs in residential dust have been positively correlated with serum levels of PCBs, but there were only a few observations that were sort of driving this correlation. And you can see on the bottom that the estimated contribution of dust as an exposure route to total PCB intake is smaller, even for children, than it was for PBDEs. So in this case, PCBs that you receive through diet are a more important route of exposure. But still, we were able to see that risk relationship between PCB levels in household dust and the risk for childhood leukemia. So whether or not PCBs are a direct marker of biological exposure, there's still some sort of indirect relationship between what's in the dust and what gets into children's bodies. And the same can be said for PAHs. So the relative contribution of dust as an exposure route to PAH is minor compared to dietary and inhalation routes, but it still could be an important route of exposure for children. Okay, so the second key question, is one dust sample sufficient to characterize contamination in a household? So semi-volatile and non-volatile chemicals do accumulate on dust particles, and typical vacuuming will not efficiently remove these dust particles that are deep inside of residential carpets. So the levels of chemical contamination that are on residential dust should be relatively stable over short periods of time at least. So this is sort of a conceptual diagram. This is not real data. This is just sort of an idea of how short-term variability of an exposure measure can impact exposure assessment. So this is a smoking history of one individual over the course of a month. And in real life, exposures are not consistent from day to day. There's a lot of variability over time. So you can see that the smoking levels from day to day are moving around quite a bit. And the problem in terms of epidemiological point of view is that in order to get a good estimate of the average level of exposure to cigarette smoke constituents, you would have to have a lot of information from a lot of different time points. You'd have to have several different measurements in order to get a good estimate of the average level of exposure. Whereas if you have something, a measure of exposure that has little short-term variability, something that's stable over time, then you could have fewer samples to characterize the average level of exposure. So if you sample on this day where there was very little smoking, your average residential dust nicotine level we would match up with the value that you got in that particular day. And if you sampled on a very high smoking day, you would still get a residential dust nicotine value that was close to the average exposure over time. So what we want to do is test whether this idea is really true. Can we actually characterize the short-term variability of residential dust nicotine? And the way that we're going to do that is by taking repeated dust measurements collected over short intervals of time and then comparing the data that we get from the same households from different sampling rounds. So this plot here is showing one particular PAH, benzo K-fluoranthene, measured in 15 residences over sampling intervals from 3 to 15 months. And you can see that there's a relationship between the level of BKF during the first sampling round and the second sampling round, and that the correlation is somewhat strong with a Spearman rank correlation coefficient of 0.76. We found that volatile chemicals such as nicotine would have more short-term variability, so the correlation between the first and the second sampling round would be less strong. And using variance ratios, we can sort of try to answer the question at the top of the page. So the variance ratio is the ratio of the within household variability to the between household variability. And what the ratios for the PAHs and the PCBs that we measured over intervals of 3 to 15 months indicate is that 1 or 2 dust measurements would be sufficient to limit the measurement error. So they'd be sufficient to characterize the contamination that exists in a household. And the difference between a PAH or a PCB that needed 1 or 2 samples is essentially how those chemicals are distributed across the population. So if you have a chemical that has a wide range of concentrations over our population, then you need fewer measurements to characterize a household. So the third question deals with the long-term temporal variability. So can we use these dust samples that are collected after diagnosis to tell us anything important about chemical contamination that existed in the past or chemical exposures that existed in the past? So again, we're a retrospective case control study. So our retrospective exposure assessment can be challenging. We want to take the information that we're getting at dust collection and say something about the critical periods of the child's development that are happening on the left side of the screen. So in our particular study, there's a latency period because we're looking at cancer. So the time between birth, for example, and the child's age of diagnosis is a median of 3.5 years. And then there's some additional time that it takes between the actual diagnosis date and when we're able to go out and actually collect the dust. So that the time between the child's birth and the dust collection can be something like 5 years. So the question is, is it plausible to say that the dust that we're collecting is relevant to the exposures that we're interested in that happened either prenatally or during the child's early development? So what we have going for us is that, again, semi-volatile and non-volatile chemicals do persist in residential dust for many years. So chemical concentrations in residential dust could represent long-term average contamination over many years. And we're hoping that levels of chemical contamination in dust are somewhat stable over these long periods of time. So again, in order to test this hypothesis, we want to take repeated dust measurements, and this time we're going to collect them over longer intervals of time. So we're looking at 200 households, and we're spacing out our sampling rounds by anywhere from 2.5 to 8 years. And then what we see is the plot here on the right. So again, on the horizontal axis, we have the concentrations of one particular PAH, benzo K-fluoranthene, from the first sampling round, and then it's plotted against the concentrations of the same PAH for the second sampling round. And you can see that there is a positive correlation between the first and the second sampling round, even though the collection dates were separated by several years. But you do notice that compared to the earlier plot that I showed, the correlation coefficient is smaller. So there's long-term temporal variability is obviously greater than the short-term temporal variability, which is logical. And we could look at this in a different way. So this is the results from the first slide with the short-term temporal variability versus that of the long-term temporal variability. Spearman correlation coefficients on the vertical axis for each of 7 PAHs that were measured in both of the studies. And the gray hatched bars for the long sampling interval, so it's 2.5 to 8 years between sampling rounds. And then the yellow bars for the short sampling interval, so it's from 3 to 15 months. And you can see that there's more correlation between the first and second sampling rounds when you have the samples spaced out by less amount of time. So the practical implication is that there's some limit to how far apart your dust sample can be from the exposures that you want to estimate before you stop getting any valuable information about what you're trying to estimate. And what we're seeing with our data is that potentially for these non-volatile PAHs, that practical limit might be something around 7 years or so. So if we can't collect a dust sample until 7 years after birth, then we're going to start getting less and less information about what happened prenatally and during early childhood exposures. But if we can sample 2 or 3 years after birth, then we'll be doing a pretty good job of estimating the exposures that might have occurred earlier. So the final key question here is, can we identify any of the sources of the variability for these residential chemical measurements? So there's at least 3 types of variability that we could try to characterize. Analytical measurement error, within residence variability, that's characterized by changes in sources of chemicals over time, so temporal variability, and then also between residence variability, that's characterized by differences in chemical sources across the population, so spatial variability. And in order to sort of try to characterize these different sources of variability, we have this sampling hierarchy where We're again sampling 200 residences repeatedly, so each one has 2 samples of vacuum dust, and then some of those samples are split so that we have analytical duplicates, so that we can compare how consistently our method is performing with the same dust from the same vacuum cleaners. And then we have an additional QC source of dust that we're running with each sample batch over the course of the study. And to look at it visually, we can split the 3 types of variability into these pie charts for the 12 pHs that we measured. So the pHs are sorted by molecular weight, so the more volatile, smaller pHs are at the top of the screen, and the less volatile, heavier pHs are at the bottom. And you can see that for most of the pHs, the analytical variability is a minor part of the total variability, with the exception of some of the more volatile pHs, which we had some issues with contamination in the lab. And it's also worth noting that for many of the pHs, the within residence variability in the green is as large, if not larger, than the between residence variability. And that has some implications for trying to measure these things with just one sample of dust. So the more within residence variability exists, the more samples you need to accurately characterize the contamination that exists in the home. So we can try to identify explanatory variables that show us where this variability is coming from. So for the within residence variability, the temporal variability, the only meaningful explanatory variable that we can find is a secular decreasing trend in pH levels over time, especially for these heavier pHs at the bottom of the list there. So you can see that still it's, even for the best case scenario, the dibenzo AH anthracene, only 15% of the within household variability is being explained by this trend of decreasing pH levels. But this is sort of a logical finding, as it seems like pH levels in the ambient environment in California are also decreasing over time. So it seems like what's in the outside world is coming in and manifesting with the dust samples. We can also try to identify the sources of the variability in terms of the between residence variability. And we had some more luck with this sort of modeling. So we've seen that residence age, which is something that Asa mentioned earlier for lead, is also important for pHs, and we've in fact seen it for all of the chemicals that we've looked at in the residential dust. So the older the home, the more PAH we're measuring in the dust. Residence type is also an important explanatory variable. So people who live in apartments tend to have higher levels of PAH. I think it's because they have smaller footprints for their homes. So you're concentrating the same sources of PAH in a smaller area, and you get higher concentrations of PAH. In the same token, carpet coverage is a predictor of pH levels. So people who have less carpet coverage actually have higher concentrations of PAH. So I think that potentially the pH levels, the pH sources in a given house, if there's less carpet to glom onto, the pH sort of accumulates in that smaller carpet area, and the concentrations are higher. Smoking habits are also indicative of pH levels in our analysis, so people who smoke have higher pH levels. In a previous analysis, we also saw that gas heating was an indicator of pH levels. And also shoe removal habits seem to be weakly correlated with PAH levels. So people who remove their shoes have lower levels of PAH, so they're tracking less stuff in from outside potentially. To sum up the sources of variability, dust is a heterogeneous matrix. So that nature of the matrix is going to lead to analytical variability just inherently, and some of that is going to be hard to get rid of. We also saw that there was substantial within residence variability in our measurements, and a lot of that was difficult to explain. And we saw that there was also substantial between residence variability, and we were able to explain some of that with residential characteristics. So let's go back and finish up by looking at our key questions and try to fill in some answers. So the first question was, are these dust levels indicative of biological exposures? Yes, especially for children, and especially for chemicals that are prevalent in dust, things like PBDEs in California. Is one dust sample, a single dust sample, sufficient to characterize contamination in a household? Yes, especially for non-volatile chemicals, and especially in what I'm calling diverse populations. So in populations that have a wide range of levels of residential dust contamination, you can have a better chance of using just one dust sample and getting useful information about the average levels that are in a home. Can dust samples collected after diagnosis be used to estimate past chemical contamination? Yes, but with the caveat that the correlation between the past and the present levels of contamination diminishes as the time period increases. So there's definitely a practical limit to how far back in time you can go with your retrospective exposure assessment. And then finally, what are the sources of variability? Can we identify the sources of variability in these dust measurements? To some extent, yes, we can. We can explain some of the analytical variability that we're seeing. We can explain some of the between residence variability that we're seeing. But as far as the within residence variability, it's not easy to characterize. So I'd like to acknowledge all the people that I work with, especially the people who are here from the CCLS group. Thank you, guys.